Hello, welcome everyone to CyVerse Learning Institute's Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer, and today's presentation is on writing and sharing computational analyses using Jupyter Notebooks in CyVerse by Peter Rose. Peter is the director of the Structural Bioinformatics Lab at the San Diego Supercomputer Center. And some of you may also know about the Protein Data Bank, which is something that Peter has helped build into an archival powerhouse of 3D structures of biomacromolecules critical for designing new therapeutic options for diseases. Peter is also an excellent teacher and a practiced webinar giver, so I know you'll refer back to this webinar today for all the nuggets of information he's going to share with you. For those unfamiliar with CyVerse, we are an NSF-funded cyber infrastructure project, and this free webinar series that we do is designed to fulfill part of our mission to train scientists on how to use CyVerse's computational resources. I'm going to take care of some housekeeping real quickly, and then we'll start the webinar. Please open your Zoom chat window and type in any questions there. Peter will address those at the end of the webinar. Today's presentation is roughly 30 minutes, and like I said, we'll do Q&A at the end. We will also post later on today Peter's slides and the video recording of this on a wiki page, and we'll send you via email a link to that information. Let's see. CyVerse is busy planning our second foundational open science skills course called PLoS and our third container camp. Both are scheduled for spring 2020, so please visit our website and for more information or to sign up for these trainings. And so without further ado, I'll turn the webinar over to Peter. Welcome, Peter, and please feel free to tell us about your current research or however you want to start. Thank you.
All right, well, thanks, Tina. Thanks everyone for joining. So we recently wrote a 10 simple rules paper. I'm sure you're familiar with that 10 simple rules series. And let's see, to get to my first slide here. So this is what I'm going to talk about today. Really, why we're trying to write good Jupyter Notebooks is really to do reproducible research. And I'll go over the motivation first. Then I'll briefly go through the 10 simple rules. But since it's a published paper, I don't go into much detail. Rather, I let you read the rules. But then I'll do a demo. And of course, we're going to do that on CyVerse to show you some of how a good Jupyter Notebook would look like and how it helps reproducible research. And then really to follow the rules, you really need to learn to use various tools. And I'm going to talk about the tools. I mentioned them, but there's obviously not enough time to actually go into any details, but there will be other CyVerse webinars, etc., that address those type of tools. So let's get started. So I mean, as you all know, it's difficult to reproduce experiments in the wet lab, but surprisingly, the same is also true for computational experiments, and it should be much easier, but it's still a big challenge. If you ever had to try to reproduce what somebody else did, often they don't, share their code, or even if they share the code, you still have problems reproducing their research. And just to explain what I mean by reproducibility and the other keywords here, because several publications have different definitions what reproducibility means. So what I'm talking about here, reproducibility means obtaining consistent results using the same input data, using the same computational steps and method, the same code, the same conditions. So reproducibility is obviously important, but for me, what is more important is actually reusability, that you can take somebody's Jupyter Notebooks and you try to create new results. You put in new input data in, you change parameters, but otherwise the computational methods stay the same. Okay, so in terms of open research, there are really four things that are important. One are papers, open access publications. Obviously, you need to also publish the code, so we need open source code. But we also need open data. We cannot, reproduce a finding without the data. But where things often break down in terms of reproducibility is the environment, the compute environment, that causes a lot of problems. And this is, for example, where CyVerse comes in, where you can set up the environment and the user, the end user, doesn't even have to worry about setting up an environment. So here are some typical barriers for reproducibility and reusability. So for example, there are different programming languages, different file formats, different operating systems, different versions of software, and all that together, if you try to run somebody else's code, you get messages like that, missing files, missing dependencies, errors, etc. So hopefully, if you follow our rules, you don't see those messages when somebody else is using your Jupyter Notebooks. Okay, I'm sure you're all familiar with Jupyter Notebooks. I guess the key is it supports reproducible research, but it's not enough. You need to also do other things to make it really reproducible. The nice thing about Jupyter is that you can combine live code with equations, with multimedia, visualizations, narrative text, so you can really tell a story, a computational narrative, in a Jupyter Notebook. And it's surprisingly popular. There are now actually 5.5 million Jupyter Notebooks on GitHub. And as you probably know, there's a next generation user interface for Jupyter. It's called Jupyter Lab. So on the top, this is the classic Jupyter Notebook view. On the bottom, this is one of my own research. You see, this is Jupyter Lab, where you have this left-hand file menu also. So today, we will actually using the Jupyter Lab. Okay, so we had a workshop here at UCSD last year, and we invited a number of faculty and postdocs and graduate students. And basically, we came up with 10 rules, and they're basically in 3 categories. One is organizing and documenting your notebooks. Then the other one is working with code. And finally, sharing that, because the reason why we want to reproducible research, obviously need to share it with the community. Okay, so I'm going to quickly go through the rules, but again, the idea is, you can read this on your own. So I'm just going to be brief and then just show you a live notebook. Okay, so about organizing and documenting notebooks. If you write a scientific paper, you have an introduction, you have the main body, and you have a conclusion. Unfortunately, if you look at many Jupyter Notebooks, they don't look that way. Usually there's some introduction in the beginning, and then there's a lot of code and a few graphs, etc., and that's about it. So really, what you want to do in a reproducible research, you want to tell a story in a notebook. It's, you have a beginning which introduces the topics. Then in the middle, you have the computation going on. You should describe all the steps. And more importantly, at the end, you should really interpret the results. You can't just show an image or a graph, but then there's no explanation. And what many people are missing, they describe what they are doing in the notebook, but they don't describe why. What does it all mean? The interpretation is usually missing. That is part of the scientific paper, but it's a good practice to put your interpretation already into the notebook itself. And then you also need to think about your audience. If you show your notebook at a lab meeting, maybe you need to show more detail, but if you show it to the general audience, you may need to do things at a higher level. So our rule 2 is to document the process, not just the results. So you should add notes throughout why you choose particular parameters, etc., etc. Let me see, I need to move this menu to the side here. Step 3 is, in a notebook, you can have different sessions and headers and so on. You should really use that. So you don't want to have long cells. So each cell in a notebook should be really limited to one meaningful step. So an example of that is if you create an image in a notebook, maybe this is one cell that creates a particular image, or one cell that defines a function. So think about it that each cell has a specific meaning. Don't put multiple concepts or calculations into a single cell. If you have long notebooks, and I've seen notebooks, they're 40 pages long, and if I see those, I'm really turned off right from the beginning. So it makes sense to split long notebooks into a series of notebooks. And then you keep a top-level notebooks that is basically a table of content that will link to the individual notebooks, and I'll show you an example of that later on. So there's a little bit about how to organize and document a notebook. I think this is pretty straightforward. unfortunately, most people don't do that. So hopefully you will be better and do this, especially if you publish your notebooks. Okay. So let's see. So the next one is working with code. What happens quite a lot as people, try out new things, and that there's a lot of duplication of code, either within the same notebook or across multiple notebooks. You may have like 3 different versions of notebooks, but you're having the same code in each of them. And in that case, you should really try to use function to split out duplicate code and have it defined once. So now we're coming to the important part, that is recording all your dependencies, because if you want somebody else to be able to run your code and reproduce your findings, you need to define all the dependencies. And with dependencies, I mean all the other software tools and libraries that you use. For example, if you're in Python, you may want to use pandas or matplotlib, etc., etc. So you really need to define all those dependencies. And there are 2 major ways of doing it. One is using conda, and in this case, you create what's called an environmental YML file to define your dependencies. Or if you use pip, you use a requirements.txt file, and we'll go into that later. Okay, so that's number 1. This is really what is missing in many cases, in terms of reproducing somebody's code. The next one is obvious version control, because as you work on a project, or you may work with your colleagues on the same project, you really need to keep track of your versions and make sure you don't break your own code and so forth. A good practice is to actually start with a public GitHub repository right in the beginning of a project, rather than doing it later. And then rule 7 is to really make your notebook a pipeline, so it's reusable, let's say with different input data sets, with different parameters and so on. So basically, once you have your initial notebook, see if you can make it more generic so other people can reuse it. That means, for example, all input data or key parameters, you should define at the top of the notebook so they're easily visible, and somebody can just change the input data or the parameters right there. And as I mentioned before, break long notebooks into multiple steps, where each step is a modular step within a pipeline. Okay, so finally, now that we have our code all under version control, we have all our dependencies defined. Now we want to share our notebooks, but there's one more thing we need to do. We also need to share and explain our data here. So if you have data that should go into public repositories like NCBI Ensemble or PDB, etc., so you should really try to put them in those repositories and refer by a persistent identifier such as a DOI. If your data don't fit into those repositories, you could consider more generic repositories like Xenodo, and then there are a few others that allow you to basically submit your data and then get a DOI for your data, and then you can use that in your notebook. But in many cases, if you write a publication, you have a small data set that goes with it. You can also just keep that in GitHub. As long as it's less than 50 megabytes, you can keep files of that size also in your GitHub repository. For example, you want to put all your data into a separate folder. You don't want to mix data and code together. So for example, you can create a /data folder and put all your data in there. Now, in some cases, you have very large data sets. You can't really use them. In that case, you may want to consider providing a sample of the data so people can at least run your notebooks on a sample data set rather than the whole original data. But then you can provide links to where the original data are located. Another important step is, if you have multiple notebooks and you exchange data in between, you want to save those immediate data, because if you save intermediate data after each step, it's a good way to look at the data to make sure there are no errors, to verify each step. And that also helps with other people that want to reproduce your data. They can look at each step. Did I actually get the same intermediate results, as I should be getting? Okay, so And you really want to design your notebook, well, to be explored, so other people can read it, run it, and so forth. And that requires a few things. First of all, obviously, you need a README file in your Git repository and a descriptive one. Many README files are just way too short. They don't really explain what the project is about and what it's useful for. Also important is to add a license file to your Git repository. And if you really want to have your data open, you should choose a liberal software license such as MIT, Apache, or BSD PLoS 3, for example. Because without a license, it's not clear how other people can actually reuse your code and data. So it's very important to add those license files. Now, Jupyter Notebooks have been around for, I don't know how long, like 5 years at least. But, if we really want to have long-term preservation of your research, probably in 20 years, there is probably no more Jupyter Notebooks. There's something new out there. It's probably unlikely it will still work in 20 years, but who knows? So if you really want to preserve your notebooks long-term, you may want to create a static version as an HTML file, for example, or a PDF file. So if you really do your Nobel Prize winning research and want to make sure people still be able to look at that, then try to create a static view of your of your notebooks as well. And now you want to have other people be able to actually run your notebooks. One way for them is basically to clone your Git repository and install it locally. But to enable a larger group of audience to run your notebooks, there are several ways you can host them in a cloud environment. The first one is the Binder project. This is a project that's headed by the Jupyter team themselves, and Google provides the compute resources behind Binder. And with Binder, you can add a launch button to your Git repository. So you click on it, it will actually launch Binder in the Google Cloud environment. There's no software installation required. So that makes it really easy for anybody to reproduce their research. Now, one problem with Binder is it's pretty limited in its capability. Binder is free and you get what you pay for, so to speak. On the other hand, CyVerse Vice, which is a new feature in CyVerse, is that, it's, as Tina mentioned, is an NIH-funded effort. So you have really much more powerful compute resources available. Plus, users can save their results, they can store their data there. So using CyVerse Vice has a lot of advantages. And you can also now launch Vice, and I'll show you this live, also just from a link. You can set up a link to launch a notebook in CyVerse Vice. And finally, to make it easy for other people to play around with your notebooks, you can add interactive features like menus and sliders to it. So they don't need to actually understand your code. They can just use menus to change parameters or use sliders. And again, I'm going to show you some examples. So finally, rule 10 is I hope you all become an advocate for open research and apply what you learned during this seminar and various other seminars. And really the goal for you will be to publish a fully reproducible paper. And for me, that means that all your figures, all your data tables, all your calculations, they are accessible to a Jupyter notebook, the code is deposited in GitHub. And here's a nice Twitter a tweet from one of my colleagues at UC San Diego, Brad Wojtek. He had this tweet last year. Basically, he's still publishing his PDF models, but with every publication that he does, he has a Git repository with runnable Jupyter notebooks that has all the data there, including how to process the data, analyze the data, and visualize the data. And he also mentioned this paper, the scientific paper is obsolete. Basically, the idea is, a Jupyter notebook can be like almost like a publication, but it's much more powerful that you actually have the real code, the real reproducible results, in a notebook. And I think some of the publishers are going into that direction of supporting, running Jupyter notebooks and hosting Jupyter notebooks. So I think over the next 2 or 3 years, you will see a lot of change how publishers, support Jupyter and reproducible research. So this was a quick rundown through the 10 simple rules. And I know I couldn't go into much detail here, so please read the paper for more details. But what I want to do now is this paper comes actually with some example notebooks, and I'm going to run those notebooks now on Jupyter Bice. So let's go to this Git repository that comes with this 10 simple rules paper. So this is this Git repository, and in example one folder, you see there are a number of notebooks in here. There's also a data folder that contains data that we need. So this is completely self-contained. Okay, so if we scroll down here, you see there's some launch links. Those launch binders, as I mentioned before, and let me just do an example here to launch binder. So you just click on this link here, and you see binder is launching. But today I'm not going to focus on binder, so we just let this run. But if you wait for a minute, you will see your Jupyter Notebooks come up, and any user can run them. But let me just cancel that for now, because I want to show you how we do things with CyVerse. So down here, I have created a launch CyVerse button. So let's click this button here. So one thing that's different, so I already logged into CyVerse, so that's why it comes up. So you need to put in a name for your analysis. I just use a default, and then I just say launch analysis. And it tells me up here now that it has successfully launched. And if I go to this little notification icon, it says access your running analysis here. So I'll click on this. And this may take, a few minutes to come up. So in the interest of time, I'm already launched the notebook. So I will just go, if I can, let's see. Go to the notebook. So once CyVerse launches, it looks like that. You see the URL up here. You see it. This is actually running on CyVerse. This is not running on my local notebook. So here is this 10 simple rules repository. And we have a few examples that I want to show you. If I go to example 1 here, there are a number of notebooks listed here. So by the way, if you're not familiar with this interface, this is Jupyter Lab, where you have this left-hand menu here. So you see here, we have some data here that we need to use. I put them into a data folder, and I created a description of the data. So here, you should really describe where your data are coming from. So in here, I documented, okay, there are 3 data sets that are being used here. This is where we downloaded them, and this is when it was downloaded. So this is also good practice, not just have the files there, but nobody knows where those data came from. So hopefully this should be crystal clear, where my data came from and when I got those data. Okay, so you see here there are 5 notebooks here, and a good idea is if you have a multi-step workflow to put the numbers in front, so it's obvious to a user what you're supposed to do here. So the first notebook is more like a table of contents here for what we're going to do. And as you see, I have description here, there's an introduction here. So what does this specific notebook do? As you know, protein structures have secondary structures, and there are different kinds of proteins. Some proteins have only alpha helices, we call them alpha proteins. Some have only beta sheets, we call them beta proteins. And we have some proteins that have both alpha helices and beta sheets. So the question is, can we use a protein sequence to predict if a protein is in one of the 3 classes? So that's what those notebooks are about. And to do this, it's really a 4-step process. First, I need to create a data set. So we want to do some machine learning. So first, we need to create a data set. And I don't want to do this manually. If I create this data set manually, it's not reproducible. So all the steps of creating that data set for machine learning is in this one notebook, create data set. Then the next step is using those data, we calculate some features that we want to use in the machine learning model. Then finally, in step 3, we fit a 3-state classification model here. And once we fit the model, then we actually want to use it to make a prediction. So you see, I broke up this task into 4 separate tasks. And the good thing about it, let's say I want to create my features in a different way, I can just swap out one notebook, but the other parts are still the same. So this is always a good idea. If there's some logic, how you can break up your workflow to do it that way. So let's just look at a few of the notebooks. Let's start with this one, create the data set. So let me just quickly run this notebook here. So, and this notebook, it's not a real scientific notebook. This is more like a companion to the 10 Simple Rules paper. And in here we explain a little bit, what rules we are applied here. So for example, share and explain your data. I already showed that to you. We have this data directory where we keep our files and we describe them and so on. So I don't want to explain the calculations here, but just go to the end. So another thing you see in my notebooks here, as we go through, I always print, if I load some data, I always print maybe the top 5 just to make sure, all the steps work. So it's a good idea in between to always print some intermediate results as you go through. You see here, those are protein sequences, for example, protein sequence data we loaded and secondary structure information. So here we calculated some data. So the next step is we want to calculate some features. So I'm going to run through this notebook here. All right, let's see. So, having this notebook, you can obviously, you have headers, text, but since this is like an HTML page, you can have links, you can have links to papers, links to other resources. So make sure you put in all the links here to make this really useful a document here. So, so what we did now here are protein sequences, and we created some feature vectors here in this last column here. This is a 100-dimensional feature vector for each protein sequence that will we feed now in our next step to fit a model. And you see at the end of this notebook, we are saving intermediate data. They go into this intermediate data directory. So if somebody else wants to reproduce my work, they can actually also see all the intermediate data that I'm generating. Now we're going to fit a model here. Let's quickly run this through here. And so this particular notebook shows how you design your notebook to be explored. And let me just wait a second until this completes here to show you what I mean. So here we run various machine learning models, and the default is a support vector machine model. But see, we put in a menu here, so the user doesn't need to know about how to change the code. If you want to use logistic regression or neural network instead, you can just choose from the menu here. And then obviously, the important feature about Jupyter Notebooks is to make things graphic. I mean, we can print out data here, but we can really include graphics here. That really makes it so powerful. So now that we fitted a model, we also need to explain a little bit about the model. That's what I was talking about. Not just explain what you're trying to solve, but also explain your result. For example, here we talk about what the limitations of that model are. You really need to explain this if people want to be able to reproduce their data and reuse it. So the final step then is, since we fitted the model, let's make a prediction here. Let's run this notebook here. So again, here we put in a text box. So if you're a new user, you don't need to know how to program. You can just copy and paste a new protein sequence in here, run the notebook, and then your prediction comes out here. So for this particular protein sequence, it predicts it's an alpha pus beta protein. So that gives you a little idea, an example how we use the 10 simple rules, to structure a notebook. And again, in this case, it made sense to break this into 5 steps here. So coming back to my presentation here, now to actually make this happen, you really need to learn a number of tools. And unfortunately, this isn't really taught in many universities. I mean, most of you probably know how to use Jupyter Notebook and Jupyter Lab, but to really make it reproducible, you need to learn how to use version control using Git and GitHub. GitHub is a place where you would store your source. Then you need to be able to define your requirements. And Conda is a great way of defining your environment, your dependencies. The nice thing about Conda is you can have different projects and they may use different versions of the same software, and they can coexist without interfering with each other. So for example, if you set up your requirements in Conda, then you can immediately launch your notebooks in Binder without doing anything. It will do it automatically for you. But as I said, Binder has some limitations. If you rather want to run things on CyVerse, which has a lot of advantages, it's much more powerful, you need to create a Docker container. And again, the CyVerse team, is giving also various webinars how to use Docker. And the way I got started with CyVerse is that I worked with the CyVerse team, and they helped me create this first Docker file. And once you have that example, then it's very easy to apply that to your own project. So if you ever want to get started on CyVerse, just contact the people at CyVerse, and they help you with your first Docker file. It's not as scary as it sounds. Okay, so now I talked about the tools, and let me show you how the tools actually map onto this 10 simple rules workflow. So we talked about it's important to record your dependencies. So Conda is probably the best choice and the easiest choice to do that. As far as version control, Git and GitHub is probably the go-to solution these days. As I said, there are about 5.5 million notebooks already on GitHub that tells you something. And then in terms of sharing your notebooks, make it easy to use for a user. You can use Binder if you have a small kind of problem. If you really need more production environment, you can use CyVerse Vice for that. Okay, I found this nice cartoon here that basically kind of summarizes what we're doing here. So here's Jane. She is developing some algorithms, and she wants to share that with the world. And she first creates a Jupyter notebook here. And as I mentioned, it's not just code. You need to really, include an introduction, you need to interpret your results and so on. Then you publish them in a public repository such as GitHub or GitLab or Bitbucket, et cetera. Then next step is you need to set up your environment, like a Docker file or a Conda environment. And then you can share it either through Binder or CyVerse and make it accessible to anyone in the world. So that's kind of a summary of this process here. Okay, so to learn more, that CyVerse offers various webinars and workshops. You can learn more about that. If you want to set up your own Vice application, a Jupyter Notebook application, there's some documentation online. But again, the CyVerse team can help you with that. This is where the 10 simple rules example notebooks live. So you can rerun the notebooks that I just showed you. You will see that it's completely reproducible with a single mouse button click. We also recently did a 4-hour workshop at the ISMB conference in Basel a few months ago, and this entire workshop is available online. So that explains how to use the tools like Git and Conda and Binder and so forth. So if you want to go through that, it's basically all online. You can go through this tutorial here. Okay, and then, so here's a challenge for you. You guys, are listening to me, passively listening to me, but here's a challenge. What I want you to do, the next paper you publish, make it fully reproducible. All your figures, tables, everything, all your computational results, try to create them in a Jupyter notebook, deposit your data and source code in GitHub, and then finally deploy it on CyVerse Vice or Binder or both. So if you really want to do that, I will be happy to help you. If you have started with it, I'll be happy to review them, I'm sure, and the CyVerse people will help you too. So that's the challenge. I don't want you to go out and not do anything. So this is your challenge, to publish a reproducible paper. I will be watching out for you. I know all your names. So in terms of acknowledgements, so I want to acknowledge all the co-authors here, especially Adam Rule, who did actually most of the writing of this paper here. And I also want to thank the CyVerse team. They were incredibly useful. I was probably one of their first guinea pigs to start out using Jupyter Notebook for my own research here. And that's about it. So I guess we'll see if there are any questions. Thanks.
Thank you, Peter. And what a great challenge. I love that.
Yeah.
If anybody's on the webinar today that rises to the challenge and succeeds, please contact me at you'll get my email. So we do have a couple questions, and I actually started off because Peter, when you were talking about making everything, and reproducibility versus reusability, I wondered about is there some sort of standard format for citing a reusable Jupyter notebook? Or is it something like you could add as a cell in the notebook itself that tells people how to cite it? Some people on the webinar responded saying that maybe to use a DOI, and other people said just refer or cite the publication itself. Do you have some input on that for us?
Yeah, so I mean, you can basically use a Git repository as a supplement. Most journals accept that now, that you just put in the GitHub URL, and that is basically your supplementary, material. But you can get a DOI. What you need to do is you basically take your Git repository and zip that up and upload that into a repository like Zenodo. There are others, Harvard, Dataverse, and others. So you can upload this whole repository in there and get a DOI for it. Now that way it's persistent, the code will never change. So that's one way of doing it. But there's nothing like a repository of Jupyter Notebooks, where you could get a DOI for it directly.
Got it. Okay. Let's see. Question: if you have a lot of dependencies that are not installable with Conda, what would be the best way to have them easily packaged for the user?
Yeah, so you can also, for the Conda environment, you can also use pip install. I mean, most software is available then as a pip install. You can have a pip install within a Conda environment. I mean, if people are interested, I can show you an example of that. Actually, that happens quite often. Not all your dependencies are in Conda. Then you can use pip install, and if it's not in PyPy, then you can still install directly from GitHub too. If that third-party code that you're using is available in GitHub somewhere, you can install it directly from GitHub, and you can define all of that in an environment or YML file. I have examples of that. I'm not sure if I want to go into that detail now, but the person who asked that question, maybe at the end, maybe I can show you a few examples like that.
Okay, thank you. Are there any other questions for Peter? Okay. I'll give you a few more seconds, but in the meantime, let me tell you about our next webinar, which is in 2 weeks, same time, same bat channel. November 8th, we'll do a focus forum webinar on using RNA-seq differential expression analyses using 2 CyVerse apps. One is called DE-seq2, and one is called Read Mapping and Transcript Assembly. That's going to be presented by Satish Puri. He's a graduate student here at University of Arizona and also works with the CyVerse team. So we hope to see you then. And I do not see any further questions. Sounds like Peter's included his information at the front of the presentation as far as contacting him if you have further questions. And as he said, CyVerse team is always willing and able to help. So see you guys in 2 weeks. Thank you so very much, Peter. Really appreciate your presentation today.
Sure, thanks for listening.
Okay, bye everybody.